Good afternoon, everyone, and welcome to the Aedes Using Gene Drives to Counter Zika webinar. Or perhaps good morning to those of you who are more than one time zone west of our offices here in Virginia. I think we have some of those. We've all heard about the alarm that the spread of the Zika virus in the Americas is causing. Although the virus was discovered as long ago as 1947, it's only in the past 6 months that researchers have started to suspect that the virus can sometimes cause birth defects when it infects pregnant women, in particular microcephaly, and possibly also a disabling, sometimes fatal autoimmune condition, Guillain-Barré syndrome. Relatively little is known about the details of these possible links, and an overreaction by governments could itself have serious consequences for affected countries. So it's important that responses be well considered. One way to counter the spread of the Zika virus would be to reduce or eliminate populations of its insect host, mostly mosquitoes of the genus Aedes, which also transmit other pathogens. One possible way to do this, and it's not the only way, it should be noted, is with a relatively new technology known as a gene drive. Gene drives are genetic constructs that can spread through natural populations much faster than ordinary mutations. Gene drives that drastically impair the reproduction of Aedes could, in the relatively near future, therefore become a real option for countering Zika. But there are other genetic approaches to curtailing Aedes populations. And Zach, when we get to you, I might ask if you could say a little about that to clarify the distinction between gene drives and other genetic approaches to countering Aedes. Once the gene drive genie is out of the bottle, however, some fear it could be impossible to put it back in. Yet, not using the best methods we have to counter Zika increasingly seems likely to yield very undesirable consequences. So it's a dilemma. I'm delighted that we have with us three leading experts: Davidson Hamer, MD, Boston University School of Public Health, who will tell us the latest on the effects of Zika and its spread; Dr. Zach Edelman of Virginia Tech, who's researching gene drives that could counter Aedes mosquitoes; and Dr. Sahotra Sarkar of the University of Texas at Austin, who's thought long and hard about the ethical issues that using gene drives to counter Zika might raise. Participants, please send your questions using the Q&A feature in your Zoom window while our experts are speaking, making clear to whom they should be addressed. At the end, we will pose as many of these as time allows when we've heard from all three speakers. We must, however, end promptly at 2 p.m. Eastern. So I'd first like to ask Dr. Hamer to tell us, as concerned biologists, what we know now and what we need to know about the manifestations and epidemiology of the virus known as Zika. David.
Okay. Sorry, this is the wrong part of the slide. Let me go back to the beginning. Ended up in the middle of my presentation. I would like to just briefly provide an overview about Zika virus. I think we'll talk more about the biology of the virus, but it's a single-stranded RNA of the flavivirus genus. It's very closely related to dengue, West Nile, and other flaviviruses, and this becomes an issue when we get to diagnostics because of cross-reactivity. The primary vector is Aedes aegypti, but other Aedes species are capable of transmission. This is first discovered in the Zika forest in Uganda in 1947. First human case, that was a non-human primate. The first human case was diagnosed in 1952. And then the first large outbreak was on the island of Yap in Micronesia in the South Pacific in 2007. Subsequently, there was a fairly large outbreak in French Polynesia. And then the first case occurred in the Americas on Easter Island in 2014. The outbreak that's currently besieging the Americas began more fully in Brazil, northern Brazil, in May 2015, and really spread very intensely between late December and the last few months, with more than 28 countries and territories now with established infection in the Americas, that is. And Brazil alone has an estimated 1.5 million cases. This map shows the discovery in Uganda, and then later cases. A lot of this was serologic testing done in parts of Africa and Southeast Asia, but also Pakistan in the 70s. And then they Zika virus reached the Yap Island and Guam in 2007, subsequently spread to Tahiti and French Polynesia in 2013, then to Easter Island in 2014, as well as New Caledonia and the Cook Islands, and then Brazil in May 2015, and then really since then there's been spread to northern South America, through Central America to Mexico, and then through much of the Caribbean. There are a few other areas around the world where cases have been identified recently, and actually we're in the process of reporting the first case, at least of a traveler to Vietnam that had Zika virus just today. This is going out on ProMed today or tomorrow, but we're also reporting it to the Vietnamese Ministry of Health because they claim they do not have Zika, but we have fairly good evidence that that individual acquired infection in South Vietnam in the last few months. So the distribution may be more widespread than we think or than we know. The virus cycles in two cycles, a sylvatic cycle between non-human primates and mosquitoes, and then it can then end up in an urban epidemic cycle where it goes between humans and mosquitoes. There are other potential modes of transmission. I mean, clearly a mosquito bite is the major mode of transmission, but we now know that there's a risk of sexual transmission. So far, this has only been shown from a man to a woman. And one of the reasons for this is there are now some studies being done looking at the concentration of viral load, and it appears that in blood it's relatively low. Urine, it's a little bit higher, but it's much higher in semen. And it also, there's some initial studies suggesting that it lasts in semen for as long as 62 days. There are actually a number of new studies starting to try and understand this question to determine the duration of carriage and risk associated with semen infection. Blood products have not been shown to transmit infection, but are a potential vector based on identification of the virus in Brazil and French Polynesian in blood products. And then breast milk is also a potential mode of transmission. And in saliva, again, there's not documented transmission by saliva, but there are actually relatively high concentrations of the virus in saliva and in the nasopharynx. So this is potentially another modality for transmission. This next slide shows clinical manifestations. Estimated 80% are asymptomatic. This is really data from the Yap Island outbreak that was very well characterized by the CDC, so we don't have much more data than that, and that's another open question. Incubation period is relatively short, as is common for many flaviviruses. Typical presentation involves fever, rash, which lasts about a week on average, joint pains, and then in a subset, conjunctivitis. There are a number of less common symptoms that I've listed at the bottom, but one that's coming out in some of the clinical studies, including one that we're analyzing a case series, is some patients will have very severe pruritus, very severe itching associated with the rash. And this is very faint, but this shows the macular rash that you might see in a patient with Zika, but it may also be more maculopapular. So you can see this is a more elevated eruption in a patient who had been in Haiti. I'm not going to go into detail, but basically one of the challenges diagnostically, at least on the clinical side, is there's a lot of overlap between Zika, dengue, and chikungunya infection. They're all carried by the same vector, and they're all occurring, at least in some places, concurrently. So what are the complications? Well, microcephaly is one that has raised a lot of questions, and I think that in the last month there's a lot more data suggesting that there really is that Zika is really responsible for at least some of the neurologic complications in newborns. And may also be responsible for early fetal demise, for miscarriages. There's some of the cases have shown Zika virus RNA in amniotic fluid, but also in situ in the fetal brain. And then there's some pathologic studies showing calcifications, a number of abnormalities of development of the central nervous system, as well as intracytoplasmic viral inclusions. Autoimmune disorders are rare, but there are a few cases reported. And then Guillain-Barré syndrome I'd like to say a little bit more about. So these are data that were just published in The Lancet from the French Polynesia outbreak, and this shows the epidemic curve of suspected Zika cases based on a clinical diagnosis in lighter color, and then the darker red are cases of Guillain-Barré syndrome. Oops. And so the epidemiologic curve of Guillain-Barré syndrome followed the epidemic peak of the viral infection. This is a little bit on the characteristics of it. I think one important thing is that they estimated 0.24 cases of Guillain-Barré syndrome per 1,000 Zika infections, which is a relatively low rate. But if we're having hundreds of thousands or millions of cases, that's a lot of patients with Guillain-Barré syndrome. There is a very rapid progression from the onset of symptoms to sort of the worst possible neurologic status, and the clinical presentation is listed there. The epidemic curve, so this is shifting to microcephaly, and this is basically showing the timeline in Brazil. So actually some of the early outbreaks were recognized in 2014 of patients with fever and rash who tested negative for dengue fever. But it wasn't really until 2015, several months into the year, that the first Zika confirmation was done. And there were a lot of women that at least theoretically were exposed during this time. And you can see that by week 46 there was a peak in basically many of these deliveries that occurred during time when Zika was spreading in Brazil occurred, and there was a substantial rise in the proportion of babies with microcephaly. So that was just a little bit about microcephaly. There's a lot more information, a lot of questions, but I think that the growing evidence base is that infection at some point in pregnancy, most likely first trimester and probably early second trimester, carries a great risk of harm. We don't know if later in pregnancy there's a harm. There's a lot of questions, and as a result, there's a lot of interest in having longitudinal pregnancy cohorts. Last couple slides are just on prevention of dengue, of Zika, and this slide was really originally made for alerting people to chikungunya and dengue, but we need to add Zika to it. And sorry, that was out of order. My apologies. These slides were slightly shuffled. One more comment. This lists all the potential adverse neurologic complications. There are many that have been defined, including both neurologic and ophthalmologic problems in babies. Diagnosis of Zika is by PCR, if possible, in the first week of life. The first week after symptom onset. And then after that, we have to resort to serologies. The problem is that IgM serology cross-reacts with dengue serology. And so if the dengue is negative and Zika is positive, that's helpful. But if they're both positive, it could be due to cross-reaction, and a neutralization test must be done. The problem is plaque reduction neutralization tests are cumbersome tests to do and are only available in a few laboratories, primarily CDC laboratories and then research labs in the US. So finally, last few slides on options for prevention and control. Obviously any measures to try and limit insect bites are useful. Insect repellents are probably the mainstay, but also trying to stay within air-conditioned or screened areas can be helpful. From a public health standpoint, trying to empty standing water from container, selective use of herbicides and insecticides, and then we'll talk more about mosquitoes and modification of them in a minute. These are recent travel precautions, and the big one is that women who are pregnant should avoid travel to areas with ongoing Zika transmission. Just in the last couple of days, the CDC released a modification of this based on epidemiologic data on Aedes aegypti distribution and risk of dengue. They recommend avoiding travels to elevations less than 2,000 meters because there's evidence that there's a very low to almost no risk of dengue or of encountering Aedes aegypti at higher altitudes. Pregnant women who have traveled to Zika-infected areas should be screened for infection upon return, and then male partners of women who are pregnant should use barrier methods after travel to countries with Zika, but we don't really know for how long. At least 2 months, but potentially longer. And last slide, why has Zika emerged now? These are all really theories. One is that there's been a mutation in the Asia strain, which is the strain that's being found in Latin America and the Caribbean, that has led to increased infectivity of the Aedes vectors, or that somehow leads to higher levels of viremia in humans and therefore facilitates transmission. Another theory is that naive populations, immunologically naive populations, South Pacific amplified the virus and allowed it to spread via international travel. There are definitely lots of competent vectors in the Americas. Another theory is that the areas where it has established and spread are areas where there's been a lot of pre-existing dengue fever. So a large proportion of the population already have anti-dengue antibodies, and the question's been raised, can this lead to an immune enhancement phenomena where cross-reacting antibodies facilitate uptake of the Zika virus? Wow. And I'll stop there. I think we'll do questions later.
Questions at the end, yes. Thank you, David, for that fascinating overview. Next, I'll ask Zach Edelman of Virginia Tech to explain the status of research on gene drives to counter Zika, or rather the mosquitoes that transmit Zika, I suppose. Zach.
All right. So I want to talk about some of the work that's being done in the field in regards to gene drive in Aedes mosquitoes, and we'll have to talk about some other mosquitoes as well. But largely, we ask the question, can we stop the biting using this type of technology? Can I make my slide go forward here? Okay. So we've already seen, I'll skip over this map like this. I wanted to put up one other slide showing of the almost 200 imported cases of Zika in the United States, almost half of them have been into 3 different states, Texas, Florida, and New York. So if local transmission of Zika virus does occur in this country, which has not yet, other than Puerto Rico and some of the island territories, it might begin in one of those states there. So here's an important point that I want to make, is that female mosquitoes of the genus Aedes drink blood in order to produce eggs. Male mosquitoes do not feed on blood. Okay? They're going to be nectar feeders, and they don't transmit these pathogens to humans. And the transmission of viruses like Zika is biological in nature. That is, the virus has to infect the stomach of the mosquito, escape from that, infect the rest of the body, and then eventually find its way into the saliva, so that the next time the mosquito feeds on a person, it can be transmitted again. And if those things do not happen, transmission via this route will cease. So as Tim mentioned, I want to talk a little bit about non-gene drive approaches first that still involve genetically modified mosquitoes before we get into the gene drive topics. And this is the technology that has made it through the regulatory system, at least in Brazil. And this is based on the company Oxitec and their sterile male suppression strategy for Aedes aegypti. And so after seeing success in the Cayman Islands and other places, they've done some large-scale trials in Brazil, which was published last year, where they were able to reduce the adult mosquito densities by 95% in their trial sites. And now this has been approved for use in Brazil. And they've applied for a permit to do similar types of releases to control Aedes aegypti mosquitoes in Florida. And as of last week, as you can see down here in the Federal Register, the FDA published a preliminary notice of no significant impact, of which, if that stands, they will allow these trials to go on in Florida this summer. And so how does this technology work? Well, in the factory, they have a genetic construct that produces a tetracycline repressible activator. So these mosquitoes survive to adulthood, both the males and the females. But once released into the field, the repressor is not present, and this activator can activate its own expression in a sort of infinite loop. And the accumulation of this protein eventually kills the insects before they're allowed to grow up. So this is a strategy that is, this is an effectively sterile mosquito, but not an actual sterile mosquito. That is, these mosquitoes produce progeny, but those progeny don't develop past the late larval pupal stage, not long enough to drink blood and not long enough to produce eggs. And so how this works is that they sort males from females in the factory and then throw all the females away and then release the males so that they can go find wild females. And then they'll pass on one of their copies to those wild females, and those wild females will produce no progeny. And if you do that long enough, you can suppress the population. So far where this has been tried, it has worked. and has reduced mosquito populations. But here's the downsides. You need to sort males from females in the factory. And at scale, when you're talking about hundreds of thousands or millions of individuals, this becomes technically laborious. And then 50% of everything you rear in your factory, basically 50% of your factory capacity, is dedicated to rearing and producing insects that are going to be discarded and not used. Okay? And then release rates must be in excess of your local population. So that's the technology as it stands now. So the question is, can things like gene drive and other aspects improve this? So on the road to gene drive, as we get there, I want to talk about some work that my lab did along with the lab of Dr. Jake Tu at Virginia Tech, asking the question, can we specify mosquito gender? Because as I mentioned, a male mosquito is not a threat. No blood and no viruses. So if we can have these mosquitoes all choose to be male, then there'll be no virus transmission and then, of course, no next generation either. So it works out both ways. So after a long DNA sequencing project trying to sequence male and female DNA, Brantley Hall, who was the graduate student who did this work, identified a new gene which was not in the genome assembly of this mosquito and had never been recognized before, and was able to show that this gene was present only in males but not in females. And we were able to do in situ hybridization to mosquito chromosomes and show that this gene was present as a haploid gene. It was only on one copy of a chromosome and not two, like you'd expect for a gene that was unique to a male chromosome or a Y-type chromosome. And so we embarked on a study to determine whether or not this new gene actually could cause males or females to become males. And so we were able to clone this gene onto a DNA plasmid and inject it in this needle into these mosquito embryos. And here's a zoom out of what these mosquito embryos look like under a microscope. And this is about what our needle looks like under a microscope. And so by injecting this DNA in, we could cause these embryos to express the NIX protein, and then we can assess whether or not they became males or remained females. And so after 2 experiments, it became pretty clear that here's a picture of, this is not what you don't usually think of a mosquito, but this is actually the posterior end of the abdomen where the reproductive organs are. And so the wild type male will use these kind of claspers to grab the female during mating, and a female doesn't have those. But our injected females had partially turned into males. And when we dissected them out, we found that these females actually had testes and male reproductive organs instead of ovaries, which you'd expect from a female. And so we're pursuing strategies now whereby the same repressible technology could be used to control the expression of this NIX gene that might be able to produce 100% male progeny in your factory. So instead of throwing away half, you could potentially use all of them in these same kind of controlled single generation sterile releases. But the question is, could you do even better with regards to a gene drive? Okay, so what do we talk about when this term gene drive? It's thrown around a lot, and it can be this kind of concept of gene drive can be catalyzed by multiple different mechanisms. The most common that you'll hear about is this one in the middle, where you have a site-specific nuclease, like a homing endonuclease or a CRISPR-Cas9 nuclease, that is able to introduce a DNA break in the homologous region of the chromosome. And then during the repair process, the first gene is used as a template, and you end up with 2. So 1 becomes 2 within a single generation, and then those can divide. In the next generation, it can happen again and again. Okay? And this was actually validated for homing endonucleases in the malaria mosquito in 2011 by Austin Burt and Andrea Crisanti's group, where they showed that this homing endonuclease could spread when introduced at low frequency into a mosquito cage population in the laboratory, showing that it worked. But these homing endonucleases were hard to re-engineer. So the big breakthrough came when people started using CRISPR-Cas9 nucleases to specify new target sites. And this was the paper by Valentino Gantz and Ethan Beyer in Science last year, showing that one of these gene drive constructs based on CRISPR could invade the yellow gene and convert these kind of black-bodied male flies into yellow-bodied flies. It didn't take very long for 2 other groups or the same group again, rather, and another group to develop the same kind of gene drive technology for 2 different malaria vectors, and both of which observed close to 99 or greater than 99% of progeny inheriting that gene drive construct for multiple generations, showing that you could indeed drive genes through these populations in a laboratory setting. For Aedes aegypti, there's no published reports yet of a gene drive like this, but there's no reason why it couldn't work at this point. We know we can do homologous recombination. That is, you can insert DNA of your choice into the location of the genome of your choice, and there's been several published papers on that. And so we postulate that if you linked this kind of gene drive phenomenon to expression of a gene like Nix, you could potentially drive maleness through a population. So every generation, you would only produce males. Those males would be fertile, but only capable of ever producing males. And that would go on and on and on until all you had left was males. And the last males would be very sad and lonely, and then there'd be no more females in your location. Due to time restrictions, I could only mention the most powerful and most popular forms of these gene drive. This comes from a very well-written chapter from John Marshall and Omar Akbari, talking about a lot of different types of gene drive. Some are more powerful than others in terms of how fast they invade. They vary in their stability and their confinability. And those who are interested can look up some of these things. But there's a lot of different ways to achieve some of the same goals. And so one of the really strong benefits that I see in reducing or eliminating mosquito populations is what I call fighting the devil you don't know. Sure, it will stop dengue and chikungunya and Zika virus transmission, and that's indeed wonderful if we were able to do that. But elimination of vector populations would also prevent the introduction and spread of new arboviruses that we don't yet know about and aren't yet afraid of from causing disease in humans and animals. And I'll show you one tree that kind of illustrates this point, which is illegible, but that's all right. You can see how many different viruses are listed in this tree. These are flaviviruses. And if I point the arrow down here, you can see where the dengue viruses are hiding. And over here is where Zika virus is hiding, and probably you've heard of yellow fever virus hiding up here. But what you may not know is what all these other abbreviations stand for. Here we can see West Nile and Japanese encephalitis. But many of these viruses have been identified, but they have never caused large-scale epidemic disease, so no one works on them, but could likely be vectored by the exact same mosquitoes and could invade at any time in the future. And these are the ones that we know about that exist out there. There are probably a much larger number of viruses that have not yet been identified that could also be vectored by these mosquitoes. So having these mosquitoes hang around our house when I talk about Aedes aegypti is just a recipe for what's the next one going to be. We have Zika now, chikungunya. It's going to be another one. So if you can suppress or get rid of those mosquito populations, you're doing not just yourself a favor now, but the future. And one of the most common questions I get is, well, what would happen if we did actually get rid of Aedes aegypti? And I'll just emphasize that this is an invasive mosquito. It has colonized the world in the last 400 years, and largely bringing with it suffering and death everywhere it goes. Its ancestral range is in sub-Saharan Africa, but it does not belong anywhere else in the world, and you could argue that it wouldn't even be missed from its ancestral range. Primarily, its role is to parasitize humans, and that's about it. So I don't think we'll miss it. So here's your final take-home thoughts about where this field is going. Okay, can these gene drive systems or sex-specific systems be used to combat Aedes aegypti populations? What would that long-term effect of that strategy be? And of course, all the fun questions about how to assess risk when you're talking about gene drive constructs that may not be able to be controlled. What level of risk is acceptable, given that if you had actually were able to control these diseases, how much good that would do, how much risk can you tolerate, and then how can you get rid of these things once you use them? So that's where I am.
Gosh, thank you, Zach, for that masterful overview. Lots to think about there. Lastly, I'll ask Dr. Sahotra Sarkar of the University of Texas at Austin to help us think about the ethical questions surrounding the possible use of gene drives to counter Zika, and perhaps in other situations as well, some of those other diseases that Zach mentioned, future unknown diseases perhaps. Sahotra.
All right. So I am going to be talking a bit about Zika. Let's see what's happening with the slides. Okay, good.
We're seeing them very well and clearly. Thank you.
Good. So the first thing I'm going to show is the risk of Zika spreading from the 28 regions where it's found now. This is a map that we just published in Lancet Infectious Diseases. And the interesting thing about this map is we look at the risk from two different species. Red means Aedes aegypti, and if you're seeing circles that are white or empty inside, that means that the added risk due to Aedes albopictus. The reason I'm showing this is that whereas we've been talking a lot about Aegypti, it's also the case, as was mentioned earlier, that other Aedes species may also be involved in spreading Zika. Aedes albopictus, they've found the virus in the saliva of the mosquito. Some people claim it's not definitively proven that it is a vector, but I would be very surprised if the evidence doesn't come through. Interesting difference between the two species. Aedes aegypti, as you heard, is an invasive species, but is not a very efficient disperser. And by and large, it needs more or less a tropical climate in terms of rainfall and temperature in order to survive. Aedes albopictus, on the other hand, is an extremely efficient disperser and has spread from Southeast Asia, which is where it began, all the way to the Americas, Europe. In the case of the Americas, it was spread because of water in tires that were being moved by ship. Now, the important thing that we have to realize, which was that just emphasized, is how important Aedes aegypti is as a disease vector, and not just for Zika, yellow fever, dengue, chikungunya, besides Zika and a few other diseases. One important thing that I would like to note is that there's a huge difference in the efficiency of the two vectors. And in general, Aegypti is a much more important vector than albopictus. But in chikungunya, there have been some recent epidemics in which albopictus seems to be very seriously involved. And there is a such suspicion, but not a proof, that this is because of genetic changes in the virus. And the same thing could also happen, if it has happened, could also happen with the Zika virus. That as time goes on, genetic changes might make other species very efficient with vectors of Zika. And so far, it's been found in at least 10 species of different genera of mosquito, Anopheles, Culex, Mansonia. All right. So as you heard, I'm not going to say much about the technology behind these gene drives. You've heard a fair amount about it. The question that I want to address is that when and if this technology should be used, and what the ethical problems are. And also the real purpose of my thing is to encourage public debate. We need to have a public conversation as to when and under what circumstances gene drives should be used in order to control species, or perhaps even drive some species to extinction. And there are huge regulatory gaps that remain in the United States, the European Union, and elsewhere. I would also like to note that gene drives have been proposed not just for disease vectors, but also for other invasive species, and there's no reason to suspect that they wouldn't be successful also in those cases. So in general, some public discussion seems to be necessary as to when and how these are done. Now, the one important thing about Aedes aegypti, as those of us who worked on dengue for decades know, is that classical biological control has not worked. The standard ways of eliminating mosquitoes by using pesticides haven't worked. In fact, pesticides themselves, in Brazil at least, have become important environmental and human health factors. And in fact, the most cogent suggestion about controlling Aedes aegypti in Brazil now is to improve sanitation and the water supply so people stop storing pots of water inside their houses where the mosquitoes can breed. In other words, the social and economic factors might be more important than biological factors. Now, for gene drives, there are two sorts of problems that arise. One is practical problems of is this likely to work, what might happen, and then there are deeper issues about whether or not we should drive a population and potentially a species to extinction. Philosophers would regard both types of questions as ethical questions, probably because of what they call the ought implies can principle. If it really is the case that we cannot get rid of Zika or some other disease by doing these gene drives, then there is no obligation to try to do it, and then other obligations about whether or not we should protect species should take over. The first issue, of course, are practical problems, and I'm listing a bunch that I've seen in the literature over the last couple of years in general for gene drives. Nobody has yet come out and specifically address these ethical issues in the context of Egypti, but I'm sure people will, I will, and others. The first question, of course, is that even if we eliminate Aedes aegypti, will we eliminate Zika? And that's a question where we really need to know how efficient the other vectors are, whether Aedes albopictus, I suspect it's a vector. How efficient is it a vector? And if it is an efficient vector, must they both be eliminated? And other species that harbor Zika should also be eliminated. The other question, of course, is whether or not we want to eliminate Aedes aegypti populations, whether introducing gene drives in one population will lead the gene to spread through other populations. And here there are important ecological questions to which we don't have the answer, which is the dispersal rate of Aedes aegypti sufficient for the gene drive to spread from one part of a metapopulation to other parts of the metapopulation and eventually possibly over the entire species. We need to worry a bit about this, and probably the best way to do it is to make sure that when the public discussion takes place, that disease ecologists who have worked a lot with dengue vectors happen to be part of the discussion. Then there is a question about gene drives that has been raised over and over again, which is what if, because of unintended consequences, and we will see a bunch of those in the next slide, gene drive begins to go wrong. Then what is it that we can do to try to control it? And there are technologies out there which are supposed to be able to reverse or at least rescue populations. Will it work for Aedes aegypti? As far as I know, this is not known, but the other panelists can correct me on this one. Then the unintended consequences, the target specificity might be overestimated. I doubt that this is a problem in the case of Aedes aegypti. Then there might be unintended dispersal. Unless the goal is to eliminate the whole species, then could this gene drive go back to the original areas where the species comes from, and do we really want to eliminate the species everywhere? Now, I'm not so much trying to answer that question in terms of whether we should want to or not want to. I am just trying to encourage more public discussion on the options that are available to us so that we can come up with proper regulatory policies. Finally, there is, of course, which conservation biologists and others would love to raise, there is an in-principle problem that if we are following such a strategy, really what we should be doing is to completely eliminate a disease vector species. And in fact, if we're doing that, Aedes aegypti should be one of the first species we do this to, because not only is it implicated in Zika, we know that it's a major vector of dengue, which had been spreading, and before the Zika crisis, had been the mosquito-borne disease that had been spreading most extensively in the world. And then, of course, it's also a vector for yellow fever, it's a vector for chikungunya, and all of that. But people might question whether we should intentionally eliminate any species at all. And this is a question that has been very, very extensively debated within environmental ethics since the early 1970s. What we now have is the technology to do it effectively, and do it quite cheaply. So it is important, in my opinion, that a public discussion of this begin. What are the debated issues that within environmental ethics that have come to bear on this? There's a question about whether all species have a right to exist, whether all species have intrinsic value rather than only humans, and then do we, as humans, have a right to eliminate species? I'm not saying that any of these questions have a definitive answer one way or the other. In fact, I have a hard time understanding what it means for a species to have a right and why species would have a right. Now, intrinsic value attributions to non-humans have fallen out of fashion because of philosophical reasons. The arguments for them were not particularly good, and very few philosophers buy them anymore, but there are people who do. But this is also a question that needs to be debated publicly, and what it means. Even if we say that something has intrinsic value, that does not mean we cannot eliminate it. That's the sort of argument people use for incarceration of humans who are criminals, because they happen to be they do have intrinsic value, but under certain circumstances we think sending them to prison is all right. In many states in this country, people think capital punishment is all right, and so individuals can be put to death in spite of having intrinsic value. So these are issues that we need to consider before we come up with a decision on when and under what circumstances gene drives should go. Finally, some environmental ethicists also hold that if we try to eliminate a species, we are doing something that we really shouldn't be doing. We are playing God. This is probably an influential political argument in this country, whatever its philosophical merit, and it also needs to be debated at some point. And the people who would be against eliminating Aedes aegypti would probably point out that the practice of conservation in the United States has been guided by what philosophers call the Noah principle. That is, every animal species has a right to exist because God gave Noah the duty of preserving them. This is part of the reasoning that led to the Endangered Species Act. Not all countries in the world use a similar legislative framework. The European Union, for example, uses habitat types rather than endangered species. And so the question whether species should be the unit of our ethical concern is itself something that is controversial. Note that the Endangered Species Act itself would not protect insect species that are pests. So there's not a legal issue here. There is a background ethical issue that we should have. Then, besides the question of whether species have a right to exist, we have the question of the wisdom of intentionally permanently removing a species, and whether or not something like an evolutionary potential or something like that get lost, and what happens where if we go down the slippery slope, what species do we eliminate and when do we stop? In the case of Zika, I can easily see somebody making a case that after Aedes aegypti, we should immediately move on to Aedes albopictus and eliminate them. But finally, it may even be the question we turn to invasive species and wonder as to whether or not we should not only eliminate them where they've invaded, but we are willing to live with the risk that the gene drives that are put in would go back to their native range and eliminate the species even there. In other words, the conclusions I'm drawing is that we need to develop a regulatory framework and regulate it now for two reasons. The Zika crisis is real and growing, and the technology is already there, or if not already there, as Zach mentioned, we're going to get there very, very soon, so we can't avoid this for decades, so to say. We need a public discussion of the in-principle issues. Under what circumstances can we say that a species should be driven to extinction, or are we only willing to say that certain populations should be driven to extinction, but the species should be protected as a whole? And then, of course, I want to emphasize over and over again, is we probably will need different frameworks for different types of problematic species, one for disease vectors, one for invasive species, and others for other undesirable species. That's all that I have now. Thank you.
Sahodra, thank you so much. That's a very helpful and insightful overview of a range of questions which we're all obviously going to have to deal with at some point, and some of them in the near future. I have got a few questions. Participants, please, if you have questions, use the Q&A tab that you should find at the bottom of your Zoom screen. You might have to mouse down to the bottom of your screen, bring your cursor down there to see it. There's a Q&A tab there. You'll be able to type in a question which will come to me, and I will ask as many of those as we have time for of our panelists. First one I have now is for Zach. Questioner asks, and I think Sahodra alluded to this in one of his slides as well, but this question is posed to Zach. Even if we eliminate all mosquitoes in the world, how do we know that other vectors won't become carriers of the virus? Also, eliminating mosquitoes, could that have ecologically negative effects?
So this is where the term mosquito gets very generic, and we need to talk about specific species. So we have more than 3,500 species of mosquitoes in this world. Only a few of them are important disease vectors, and none more important in terms of ovoviruses than Aedes aegypti. So if we eliminate Aedes aegypti from the world, would other vectors become carriers of the virus? And as you've already heard, other vectors are carriers of the virus, but they're just not as good. Would they become better? Whatever they're going to do anyway, because there is no contract between Aedes albopictus and Aedes aegypti to split up humans and only work out some kind of backroom deal. So as Aedes albopictus wants to feed on humans, it will, whether or not Aedes aegypti is there. And in fact, if you look at the history of the invasion of Aedes albopictus in this country, it actually has displaced Aedes aegypti from a lot of different portions of this country without any intervention by humans, just through its own competitiveness. So as other mosquitoes come in, they're going to push each other out. They'll compete with each other. So the question is there a mosquito that's more dangerous than Aedes aegypti? And there's certainly no evidence of that in the world. If one will eventually become that dangerous, well, at least we'll have gotten a break from, and maybe have eliminated these viruses from the world. And then we'll deal with that at that time.
Sure. Thanks. Related question here. I think also for you, Zach, what's the evidence that other mosquito genera, Culex, Anopheles, etc., can transmit Zika?
I haven't seen that evidence yet.
Fair enough. Thank you. Some more. A provocative question is provided here, and I think I know the answer to this, and I think it's no, but I'll ask you or any of the other panelists. Genetically sterilized, in quotes, Aedes mosquitoes with an observed offspring survival rate of 5% were introduced into Brazil to try to control various diseases caused by Aedes. The questioner asks, to what extent is it possible that the Zika virus mutated because of mutations in the host introduced by this engineered mosquito, and that this mutation may have led to infection of Zika by Zika in pregnant women, increasing the likelihood of microcephaly? What do you think of that?
Was that a question for?
I think I'll ask David to tackle that first, actually, if he would. I'm not hearing you, David. Okay, we are.
I was on mute. I'm not sure I know the answer to that question. This is part of it. Could you repeat the question again?
Okay, the questioner is raising really the provocative suggestion that the genetically sterilized Aedes mosquitoes introduced into Brazil might have selected for, or I suppose, encouraged the spread of mutations in the Zika virus that caused it to become more pathogenic. In other words, that what we're seeing is an unintended consequence of the use of these sterilized mosquitoes.
That's an interesting speculation. I don't think there's any data to support that yet. I mean, I think I think that's quite highly speculative, and you know, there's really nothing that I've read, but I haven't really been following entomology closely to suggest that might be the case. I think there are a lot of other factors in play that you know, allowed Brazil to be an ideal sort of stomping ground for Zika to enter and spread, because of the abundance of effective vector in the immunologically naive population. And potentially the fact that a lot of people have had prior exposure to dengue, if there is some form of cross-immune enhancement. Mm-hmm. I don't know that you know, I think time will tell whether that theory is what you just described is plausible or not.
Thanks. I believe I'm right in saying, that there is, retrospectively looking back, some evidence that Zika caused microcephaly in Micronesia, in the Micronesia outbreak. Is that right?
That's correct.
Yes. If that's the case, surely that argues against the speculation that this questioner put, because the genetically sterilized Aedes mosquitoes were not introduced in Micronesia.
Correct.
So we seem to have some evidence against this suggestion. No, that was my thought when I first read the question.
I agree.
Right. Okay. Thanks for that. Another question. No control methods are 100% effective. There'll always be some individuals that remain uncontrolled by this technology. The remaining populations could have been through an evolutionary bottleneck and may have been very different than the former populations. They may be resistant to this technology and possibly a far worse pest. Are there, therefore, plans to back up this technology with other control technologies like pesticides to reduce the chances of producing resistant vectors? And that one, I think, is for Zach. I guess we can see an analogy here with overuse of antibiotics.
Sure. And it would be clear that if these technologies are deployed, they're going to be deployed and the pesticide treatment programs will not stop during these processes. And actually, the same kind of bottlenecking would occur during pesticide treatment, where we see pesticide resistance emerging from those few individuals. But does that give them new traits in terms of disease transmission? There's no evidence of that. And certainly, if you're using a suppression-based approach based on maleness or mating, then your resistant individuals would be resistant to mating. And so they may not produce progeny either. And if they did acquire a genetic change that allowed them to mate, but only with themselves, but not with your strain, it would be fairly easy to introgress that in a laboratory environment and reintroduce it. So that's something that people look at. Nothing is going to be used by itself. We're not going to stop every other control measure. Everything else will move forward at the same time. This would be one more tool for the toolbox if people chose to use it.
Okay, thanks. David, again, I think the spread of Zika mainly seems to have been within tropical or subtropical regions. Are there any temperature limitations on the virus that might limit its spread in temperate regions? And let me mischievously throw the idea of climate change into that and how that might play out in future.
I think the temperature limitations currently relate to the vector. So the primary vector, at least for the outbreak in the Americas, has been Aedes aegypti, and Aedes aegypti has limitations in terms of temperature and humidity range that it can replicate within. Aedes albopictus is hardier; it can stand cooler temperatures. So, if it is shown to be a competent vector, there's a risk of a more wider distribution. I'm not aware of any data that suggests that Zika virus itself has temperature limitations, but the reality is, it probably stays warm within the body of the mosquito and within the human host. So, as long as it can go from one to the other effectively, the external ambient temperature may not affect the virus so much, but it does affect the vector.
Right, I understand. Thank you. Question for Sahotra. Sahotra, you spoke about the need for public conversations around these issues. But are those public conversations happening? Are there proper institutional forums for those conversations to take place, in your opinion?
The conversations have not been happening to the extent that they should be happening. There have been about 4 papers that have published asking for discussion that would lead to the creation of protocols about when gene drives can be introduced into wild populations, and a lot more probably needs to be done. In fact, I and several others are trying to get a group organized that will, in one of the meetings of either the Society for Environmental Ethics or in the American Philosophical Association, raise these issues and then take it to a wider body of academics beyond the philosophers who can actually advise on creating public policy. So they've begun, but have not been happening to the extent that they should be.
Okay. And I think I'm right in saying that the National Research Council has a committee looking at
yes, they do
drives as well. Yep.
Yeah.
So there are some prospects there. Question for Zach. Can you clarify the status of your research now on using gene drives to control NICS expression? Are you actually doing this, or are you waiting for somebody's approval to do this?
Now, what we're doing right now is starting to walk before we run. So we're working on these kind of repressible NICS expression vectors for doing sterile programs that involve all the males as a first step. And if that moves forward and goes well, we may incorporate a gene drive component into that, but obviously this will slog its way through the regulatory system like anything else would. So there's nothing imminent. There's nothing that no one's acting rashly in terms of Zika, and in fact, it's unlikely that any of these gene drive scenarios are going to be of any help in Zika in particular. But what we know and what we've learned from enough of these viruses is that there's going to be another Zika, different virus, 5 years down the road, 3 years down the road, 7 years down the road. So the question is, can we have these technologies ready before the next one hits? Because there's going to be a next one and a next one. We know that they're out there, and we know that the mosquitoes can vector them.
Okay, thanks. I'm surprised to hear you say that. I had imagined that we foresaw the Zika epidemic lasting for a number of years, and that we could have gene drives to counter it within a few years. But you think not?
I don't think so.
Okay, thanks. We got a question, another one for you, Zach. Do you believe microfilarial nematodes have a role in enhancing the transmission in the mosquito?
Yeah, that's an area that there are some people working on that I'm aware of, not just in terms of Zika, but in terms of any kind of interactions between filarial parasites or other kinds of infectious agents and how they affect vector competence and virus transmission. It's a really interesting and understudied topic, and we need more data there.
Okay, thanks. Last question, I think, and it will have to be for Sahotra. What may be ethically wrong in eliminating an invasive species from areas where it did not exist before?
The worry here is that the worry here is not so much a question of eliminating an invasive species from a new area, but whether or not we want to use a technology like gene drives to do so, where there is always a possibility that the genes that are responsible will go back to the original area in which the species is found, that is to the native range, and also spread there, and we might end up eliminating the species as a whole. That's the real worry, and that relates to the worry as to whether we have a right to eliminate species as a whole. The other thing we should also remember about invasives is that for all of the dislike that people have for them, a lot of the invasives, especially ones that have been there for generations, may well be playing an important ecological role in the new areas in which they have spread. So we need to be careful when we move into a situation like that.
Understand. Thank you, David, Zach, and Sahotra, for sharing your insights into this alarming problem and possible ways forward. I regret that we don't have time. We've got more questions lined up. I regret we don't have time to deal with those. If people would like to email questions to me, tbeardsley@aibs.org, I can try to have them answered and later. That concludes our webinar. Thank you to our expert panelists. And on this closing screen, you can please find details of the next webinar in the AIBS Leadership in Biology series. Good afternoon, everyone.
Thank you.
Thank you.
Thank you.